My name is Tom Grabowski, and I'm as Jeff mentioned, in the relatively unusual position of being a neurologist, primarily appointed in a radiology department. And that is because my research work has been in MRI functional imaging. I'm going to bring that perspective to this talk to tell you a little bit about how we've organized our imaging resources for the ADRC. So here are some of the things I'd like to touch on. A little bit of background. Some of this you saw already in your background video preparation, but I thought there were a couple points really worth emphasizing, specifically around imaging biomarkers. We'll talk about how the research themes that we have in our Alzheimer's Center affect the way that we pursue imaging here. I'll say some words specifically about the imaging and biomarker core, and then what are some of the specific research resources available to you and to our other colleagues at UW. So just as a starting point, everybody is aware that there are several hallmarks of Alzheimer's disease, and that is neurodegeneration, which is, I'm going to try to get a laser pointer version here. I don't see it. Neurodegeneration, which you can see in the atrophy of the cortex in this gross pathology specimen of someone who passed away after living with Alzheimer's disease. And then there are the pathological microscopic hallmarks, amyloid plaques, and tau neurofibrillary tangles. And what I want you to take away is that these three features, amyloid, tau, and neurodegeneration, are not only the hallmarks, but are the features that we seek to be able to measure with imaging as biomarkers of Alzheimer's disease process. And just to say that one reason imaging is so valuable in Alzheimer's disease, as in all neurodegenerative diseases, is that these conditions are not truly diffuse processes. They unfold in a way that impacts some brain regions early and hard, and others later, and some maybe not at all or very minimally. And this is illustrated here by the sort of standard way that we stage the neurofibrillary or tau-based pathology of Alzheimer's disease with 6 successively severe Braak stages that begin with involvement of the medial temporal lobe, entorhinal cortex, hippocampus, and so forth, and then unfold spreading through the rest of the limbic system and the adjacent ventral temporal lobe, and finally to neocortical regions. And this sort of progression in space and time is something that is super important to understand when approaching Alzheimer's disease, and it's one of the reasons that imaging is so valuable. And in general, these Braak stages correspond to clinical stages of disease, that is prodromal, mild cognitive impairment stages, when the involvement is medial temporal and limbic, and dementia only as the isocortex is involved. The roles of MRI and PET imaging are fairly distinct. MRI is predominantly a structural modality in this context, really looking for the presence and pattern of neurodegeneration that we can use, to support a diagnosis of Alzheimer's disease, and maybe most importantly, exclude other processes that might show up on a structural image. And I can say more about that if you're interested. And then as a progression biomarker, are things getting worse and at what rate? In contrast, PET imaging is what we use to obtain functional information, that is information about the metabolism in the brain. And I'll say a word in a minute why that would be important. And for its molecular specificity, that is its ability to trace particular molecules in the brain, in this case, amyloid beta and paired helical filament tau. So again, PET can serve as a diagnostic biomarker. If we see a characteristic pattern of hypometabolism for Alzheimer's disease, or if we see uptake of tracer that indicates that the molecular hallmarks of Alzheimer's disease are present in the brain with amyloid or tau ligands. And it can also serve as a progression biomarker. And just, this is probably the progression biomarker that's received the most attention, and that is measurement of hippocampal atrophy with structural MR. You can see how the hippocampus should fill the temporal horn of the ventricle, and how that the atrophy is pretty evident even at the amnestic mild cognitive impairment stage of disease. And then you can see that spread through the adjacent temporal neocortex here as Alzheimer's dementia takes hold. And we're able to use quantitative imaging techniques to segment the brain into its component structures using a three-dimensional T1 weighted MR image that Swati may say more about, and project hippocampal volume and anterior temporal horn volume onto a normal sort of normative database. And you can see excursions here below the 5th percentile for hippocampal volume, greater than the 95th percentile for inferior lateral ventricle volume, and the ratio is something we call hippocampal occupancy, and a very sensitive measure of medial temporal atrophy in early Alzheimer's disease. Likewise, we can use MR to measure cortical atrophy in Alzheimer's disease, and we've gotten very good at using the same sort of three-dimensional T1 weighted image to measure thinning of the cerebral cortex. And as I hinted earlier, that's going to appear in some brain regions more than others. This is work that is pretty well established at this point, and there's a pattern of regions, pattern of mostly association brain regions that reliably atrophy in Alzheimer's disease, and have been called the sort of MRI signature, if you will. It's a sort of composite region of interest that is sensitive to Alzheimer process. And at the group level, you can use thinning in these MR signature regions, even in preclinical epoch, to stratify participants in a study into those who are likely to cognitively decline and those who are not. And so that's illustrating the use of MRI structural imaging as a diagnostic and progression biomarker. The problem is that this isn't all that sensitive at the individual level. It's really works at the group level. So here's a typical MR image in an Alzheimer's disease patient. And now that I've sensitized you to the sort of pattern that you should expect to see, here it is. Here's the medial temporal atrophy, and here's the lateral and medial parietal atrophy, so characteristic of Alzheimer's disease. But the problem is that this sort of central tendency is obscured by several factors. One is that not everybody develops this pattern of medial temporal atrophy followed by parietal atrophy. Here's a case of Alzheimer's disease in which there's prominent medial temporal atrophy, but there's really very little parietal atrophy. And here's another case in which there's very little hippocampal atrophy, but super prominent parietal atrophy. And we've learned that there's heterogeneity in the way Alzheimer's disease unfolds with these variant presentations, which here are the limbic predominant presentation and the hippocampal sparing presentation, which account for substantial numbers of cases. So collectively, they probably account for 1/4 of the cases. So there's a lot of, there's some variability in the way this pattern actually unfolds. The second reason things are, second reason we shouldn't be satisfied with MRI alone in this field is that the Alzheimer spectrum unfolds over a very long period of time, and there's this long preclinical latent period of disease, and the MRI atrophy is gradually showing up through the latent and amnesic MCI period. And you can see here on this plot of the canonical sequence of Alzheimer's disease biomarkers that really the MRI is abnormal only about half the time by the time someone is well into the mild cognitive impairment stage. This is just not sensitive to the early process. And the third reason that it may be one thing that sort of accounts for this variability is that individuals have varying abilities to tolerate Alzheimer pathology in terms of whether or not they develop symptoms. And this is, for those of you who are connected with the center in any measure, you're aware that resilience to Alzheimer's disease is a major area of interest for us. And by resilience, we mean exactly that there's this variable relationship between function and burden of pathology. And imaging is sort of a key way to help think about this. I'm showing you here a graph of where you were using what's called the residual method to estimate resilience. And so the way you do this is a regression of some measure of disease impact on some measure of cognitive function. And of course, you'll be able to fit a line to this. And some individuals will beat this in terms of having better cognitive function than you would predict given their degree of brain atrophy. And some will fall below the line and have worse cognition than you would predict given their degree of brain atrophy. And if you take this residual that I just articulated and you correlate that with years of education, for example, or some other lifestyle factor that we found is contributory to building this sort of resilience of brain reserve, you'll see this positive correlation. So these are data that are taken from our Memory and Brain Wellness Center clinic, just clinical data. And you can see that residual is a function of your educational background. And so this variable degree of resilience contributes to the difficulty in interpreting an MRI image in the context of mild memory complaints, because you have varying degrees of resilience, you have this long preclinical period of disease, and then you have the topographic variability of the disease as well. So you'll frequently get a very tentative interpretation, or maybe very conservative interpretation of what that MR image actually signifies diagnostically. So we've just gone over that 3 reasons that structural MRI is not particularly sensitive and specific for early AD degeneration. So this is why we use PET modalities in many cases. So metabolic PET with FDG or fluorodeoxyglucose as a tracer is a more sensitive measure of neurodegeneration than MRI. In other words, metabolism in tissue will decline before there is structural atrophy. And I'm just going to illustrate that here with just a case from our clinic. So top tier here are flare images. This is a species of a T2 weighted MRI scan. And this is read as essentially normal, not showing atrophy. On the bottom tier are FDG PET images in which the tracer is, the density of the tracer is high if the signal is dark. And what you should see when you give labeled glucose analog into the blood is this avid uptake of the sugar into the brain, because the brain wants to burn glucose for fuel. And you should see this nice even uptake around the rim of the brain, but then you get to the cold spot here in the left parietal lobe in this particular patient. And if you now go with the advantage of hindsight, you can probably talk yourself into there being a little bit of atrophy on the left versus the right parietal lobe here, but it's subtle, and it's not something that is going to be identified with any degree of confidence in the clinic. So here is a pattern of hypometabolism. And we typically display this clinically with a software tool called Cortex ID that projects the signals onto the brain surface and then z-scores them against a normal database. And so in this lower right-hand image, you'll see a heat map where hotter means more hypometabolism, more profound loss of metabolism, and highlighting in this case, a left hemisphere temporal-parietal reduction in metabolism. That makes sense. And so we arrive at, there's an MRI signature of Alzheimer's disease atrophy, and there's an FDG PET signature of Alzheimer's disease metabolic reductions, which in its kind of full-blown state is seen here. And then there are two other PET modalities which are being employed, the amyloid PET scan. So amyloid PET had really revolutionized the study of Alzheimer's disease by being the first way to really certify diagnosis in life of Alzheimer's disease. I'm showing you 4 amyloid PET scans here. Inside the 2 green boxes are normal scans, and inside the 2 red boxes are abnormal scans. And they have the corresponding structural MR images are along the bottom tier. And I'll explain this to you, and then it'll be obvious to you why these are normal and positive scans. The thing you need to know is that the amyloid tracers have this non-specific affinity for the white matter. And if you look at these normal scans, you can imagine, you can see the white matter skeleton of the deep white matter skeleton of the cerebral hemisphere here. And you can see little fingers of low tracer uptake penetrating because these are the cortical folds. And they should not be taking up tracer because there shouldn't be amyloid deposited in those cortical folds. I hope that makes sense. If you look at the 2 images in the red box, you'll see that you cannot, in most places, distinguish the white matter from the gray matter compartment in the brain. And this is because the tracer has been taken up specifically into the amyloid deposits in the cortex and obscured that gray-white junction. And the interesting thing is that by the time people have any symptoms, any memory impairment, this is not a close call. The amyloid scan, if the problem is due to Alzheimer's disease, that amyloid scan is going to be positive. And if it's not, it's going to look like one of those scans on the left, and it's very rare to get something on the borderline. The second kind of molecularly specific PET scan that we do is tau PET. The tracer that we are planning to use here at University of Washington is this one called MK6240. There are several tau tracers. They all basically do the same thing, which is to home in on paired helical filament tau, which means that they are going to find neurofibrillary tangles. And you'll see tracer uptake that'll look something like this lower tier image here, in which basically you recapitulate that Brock staging that I talked about at the top of the talk. So we have, going back to the idea of imaging biomarkers, so we have biomarkers for all 3 hallmarks of Alzheimer's disease. Neurodegeneration measured with MRI or FDG PET, the amyloid component that's traced with the amyloid PET scan, and the tau component, which is traced with the tau PET scan. These amyloid and tau PET scans behave very differently. The amyloid scan, which detects amyloid plaques, pretty much will be maximally positive already by the time of diagnosis. In fact, the scans begin to turn positive 15 years in advance of symptoms. And the pattern of amyloid deposition in the brain will look very similar regardless of the clinical state of the patient. So whether they have mild impairment or advanced dementia, whether they evolve in the classic amnestic pattern of dementia, or if they have a variant presentation, the amyloid scan would basically always look the same. So you don't get really much spatial information from an amyloid PET scan that's useful. But what you do get is really this ability to certify diagnosis in the preclinical epoch and to certify diagnosis at the time of presentation. So it's, for example, just the mainstay of clinical trials, to make one eligibility criterion a positive amyloid scan. In contrast, the tau PET scan is remarkable for the variable distribution of the tau. I said a minute ago, it'll recapitulate the Braak staging. It's very sensitive to the state of the severity of the disease in the patient, and especially to the variability and the pattern of cortical involvement of the tangles. So we'll get to this a little bit. Maybe I've touched on it a little bit with that hippocampal sparing and limbic predominant species of Alzheimer's disease. The tau pattern in those two conditions will be different. And for that reason, you do get a lot of interesting spatial information from the tau PET scan, and it's also very interesting as a potential progression biomarker. Okay, so amyloid, tau, and neurodegeneration, ATN, is the kind of label for the new National Institute on Aging Alzheimer's Association research framework for diagnosis of Alzheimer's disease. This is the standard to which the Alzheimer's Centers program is now aspiring for research diagnosis. And it's just like it sounds. A stands for amyloid. You can get a measure, a diagnostic measure of amyloid, or a diagnostic biomarker of amyloid from amyloid PET. You can also get this from spinal fluid testing. Likewise, for tau, you can get a diagnostic marker from tau PET or from spinal fluid. And neurodegeneration, which you can define from some MR approach, either hippocampal atrophy, Alzheimer's disease cortical signature composite ROI atrophy, or also from some spinal fluid measures like elevated total tau. And then this basically the three binary decisions, do you have amyloid, do you have tau, do you have neurodegeneration, can stratify anybody, any comer, into one of 8 boxes here. And in this framework, if you have a positive amyloid marker, you have Alzheimer's disease spectrum or continuum by definition. And if you also have a positive tau marker, then you have Alzheimer's disease, full stop. And this is the biological definition of Alzheimer's disease. And you'll notice that I didn't say anything about cognition there. So you can have Alzheimer's disease causing mild cognitive impairment, or you can have Alzheimer's disease in preclinical stage, but you do have Alzheimer's disease. And this just to illustrate that you can use just imaging if you want to classify people on this ATN system. So on the left is someone who's amyloid positive here, tau negative, no neurodegeneration. This would be a typical preclinical Alzheimer case. Might be 10 or 15 years in this stage before neurodegeneration occurs. On the right, someone who's amyloid positive, tau positive in the posterior association regions with brain atrophy, and this would be a typical Alzheimer's disease symptomatic scan pattern. And this work with these biomarkers has really been really the culmination of years of work, largely supported by something called the Alzheimer's Disease Neuroimaging Initiative, or ADNI, which is pretty much a household word in the imaging community. ADNI has been going on since 2004, has supported a study of thousands of participants at this point with, with progressively better standards of brain imaging, and has been particularly significant as setting a standard for open neuroscience in the field. ADNI has supported like an extensive amount of scientific work by making all of the data freely available to the scientific community, as soon as it's quality controlled. And there are those of us in the audience who have published papers with ADNI data, and it's been super impactful, and has basically set the standards for how data should be taken, especially with structural MR and some of the PET modalities. And this has now been formalized in something called the Standardized Centralized Alzheimer's Neuroimaging Project, or SCAN, which was just funded this last year, and will now, there have been kind of big incentives put in place that all the Alzheimer's centers will take their imaging data according to standards, which are really ADNI standards, if that makes sense. So that'll bring me to the UW ADRC. So the title of our P30 application was Biological Heterogeneity of Alzheimer's Disease, and it really signifies the primary interest of our center in the mechanistic and biological underpinnings of Alzheimer's and the factors that counter degeneration and dementia, that lead to resilience and resistance to Alzheimer's disease. We're also very interested in early disease, variability of presentation, and novel biomarkers for early disease, vascular factors in Alzheimer's disease and related disorders, and Swati will talk about that. We have a secondary but important interest in frontotemporal lobar degeneration, and then a nationally significant program in Alzheimer's disease in American Indians. And so our resources to sort of support this work are organized in sort of shells of progressively deeper characterization of patients. We are pulling our participants from recruitment pools that have thousands of potential participants, especially the Memory and Brain Wellness Center clinic at Harborview, where the sort of theme is early detection, and we have a full-time embedded recruiter and a resulting registry of people who are ready to participate in research that's about pretty close to 700 individuals at this time. From those recruitment tools, we put together a longitudinal observational cohort that's about 400 individuals. It's a little lower than that right now, but we're building it up to 400 and aspire to 500. That you might hear us refer to colloquially as the clinical core. And these are the flagship participants in the center that are characterized according to national standards. Something called the Uniform Data Set, which all the Alzheimer's centers take to characterize the cognition of their participants. Imaging according to ADNI standards, as I mentioned. There are standards for how to do autopsies in these patients and so forth. And in our hands, one of the defining organizational principles here is really to understand biomarkers over space, because we're really interested in this clinical heterogeneity of Alzheimer's disease as a clue to the underlying mechanistic heterogeneity. And then that's the sort of clinical core cohort. And then inside that shell, there are smaller groups of participants that are characterized a lot more extensively. And these would be the folks that would go on to have, for example, tau imaging or induced pluripotent stem cell lines, single cellular omics, and so forth. And then kind of inserted there, we have had a satellite effort where we're trying to bring American Indians and Alaskan Natives into the mainstream of Alzheimer's disease research. So this imaging and biomarker core that supports our research themes is new to this P30 instance of the ADRC, which started last May. Some of the decisions and design features here are that we draw from these large recruitment pools. We're doing systematic multimodal MRI because we're really trying to characterize heterogeneity of biologic process in our participants. This includes support for vascular imaging projects, which are relying on our clinical core participants. And Swati will tell you more about some of that. I've mentioned our interest in subtyping variants of Alzheimer's disease and the relevance of tau PET to doing that. And so we really are using tau PET in preference to amyloid PET in the center. That's a sort of a resource decision. You know, if we could wave our magic wand, we would do it both in everybody. But these are very expensive modalities, and doing a PET or either tau or amyloid is about $5,000. So it's actually a lot more efficient for us to get our amyloid biomarkers from spinal fluid and reserve the scan money for the tau imaging. We have this interest in resilience, and I told you a little bit about the residual method for defining resilience, which means we need imaging measures. And then we have this systematic integration of our imaging in vivo with neuropathology, which is mediated by an ex vivo or postmortem MR scanning project, which is really pioneering. And I wish I could say more about it, but Dr. McDonald, the co-leader of the Imaging and Biomarker Core, has really put together a remarkable effort there. And so we have these in vivo measures that we're taking with MRI and PET modalities, giving us all sorts of measures of biologic processes that have to do with the structural integrity of the gray matter and the white matter, for example, the physiologic integrity of the small blood vessels, and the presence and distribution of amyloid or of Alzheimer's disease biomarkers. And we're computing a lot of these measures over a set of brain regions that we also plan to sample at autopsy. So there's this kind of large, you know, as far as Alzheimer's centers go, fairly large roster of cortical target regions that collectively embrace the spectrum of variability of degeneration that we see clinically. And so we're systematically collecting imaging and pathology data at those points systematically in the center. Similarly, we can measure a lot of aspects of degeneration with postmortem MRI. And the advantage of the postmortem MRI, it is the state at which the brain was at the time the autopsy was done. So we have really nice procedures now for registering in vivo, ex vivo, and neuropathology data, and are building a really interesting database from that. So that's basically what the Imaging and Biomarker Core is about. You know, getting MRI scans from basically all participants in the center who are either normal cognition, mild cognitively impaired, or early or mild dementia. And we're building this sort of topographic cohort, if you will. We're systematically getting ex vivo MRI data in all decedents in the clinical core. By the way, these same procedures are being done for all decedents in the ACT project and some other related projects. So there's a lot of crosstalk of these projects, especially at the level of the neuropathology. I'm not going to dwell further on it here, but we're also to do the ATN classification for all the participants in the center and sort of provide the expertise for how to understand these differing profiles of degeneration and resilience and how to operationalize these methods for different people's projects. So if you were interested in using some of these resources, there are several really good databases to approach. The Memory Brain Wellness Center Clinic itself has a data repository of all of the patients in the clinical center. That's more than 10,000. We have done a lot of work to really curate the MR images from about 500 of them. So that's a really nice resource for generating hypotheses that you might want to test in a more rigorously obtained data set later. Like I mentioned, we have about 700 potential participants in our research registry. These are people interested in research and recruitable into projects. Some of them are recruited into the clinical core. which we maintain at a strength of about 400 participants. These are really nicely characterized participants with genetic, cognitive, imaging, spinal fluid in almost all cases, or we aspire to that in almost all cases. And also acquired to the same standard as participants in other Alzheimer's disease centers. So you potentially could pool data across centers if you were interested in this level of the data. And then we have these smaller subcohorts that we're developing, the topographic cohort that I mentioned, the resilience cohort, a cohort that would be focused on vascular imaging. And then finally, the satellite American Indian Corps has, independently of everything I've just talked to you about, its own project with about 450 American Indians that have had a lot of cognitive characterization and MRI imaging on 2 occasions. There's a lot of resources out there that are that we're making available to the local scientific community. And you might use these in several ways. You might use our expertise just to help you design your study, especially if you were applying for a development project in the center. We can help you access data at these different levels, at the level of the clinic repository, at the level of the longitudinal cohort, or we can help you access public databases like ADNI, or like the overall NAC or SCAN database, which is being built up for addressing questions that you might have. The participants in the clinical core are in general available to be recruited for other studies, so we can provide well-characterized participants for studies that are initiated at the University of Washington. And in some cases, these studies integrate and even help drive the composition of the clinical core. And here are some of the folks that we have. My co-lead, Dr. McDonald from the neurosurgery department, Dr. Rane, whom you'll hear from in a minute, and my colleagues in the Integrated Brain Imaging Center, especially interested in vascular imaging approaches. And then we have consultants that are involved in the informatics and biostatistics of this, Dr. Mooney, Dr. Shojai, and Dr. Terry is an expert in applying PET technology in neuropsychiatric disease. So I think that's my last slide. I'll maybe stop and turn it over to Swati, and thank you for your attention.
Fantastic. Thank you, Tom. We'll move on to Swati, and we will hang on to questions as people have already started to enter them into the chat. So go ahead and keep doing that. And then we'll just kind of try to organize them at the very end as we open it up for questions.
Can you see the slide? And can you hear me?
Yep, looks great. Sounds great.
Okay, perfect. So we all heard from Tom in detail what imaging at the UW ADRC looks like for both MRI and PET. This presentation will focus more on just the MRI and how the imaging and biomarker core and researchers in the core have used MRI in the select maybe 100 participants to answer different sort of questions. And you will see why I say different sort of questions. So I won't belabor too much on this, but we all know Alzheimer's disease, like Tom mentioned, and we've heard in previous talks, it is characterized by accumulation of amyloid plaques and tau. When we think of biomarkers or a series of events as they occur in the elderly, as they move from a relatively cognitively normal stage to a dementia stage, we can detect increase in amyloid, then we detect changes in tau. At some point, this is the orange line is where MRI comes in, and we start seeing the hippocampal atrophy. And then we see some clinical features like memory dysfunction, and then you have full-blown dementia. If you think of MRI, MRI comes in here at this MCI stage. It is quite late, like the Alzheimer's processes have started much before that. And Tom did allude to that a little bit. And I'll just spend a second here. PET measures the pathological hallmarks of AD, A beta 42 and tau. So why do we really need MRI? And from Tom's presentation, it was amply clear that MRI cannot do this. It's not diagnostic. it can look at atrophy. And that's it. It didn't figure out even in the ATN classification. A and T are the most important parameters. But we do still have a lot of challenges with AD. We know that the number of elderly with AD pathology are going to increase and triple by the time it's 2050. We don't really have a good standard of care to test the general population. So think of doing PET in the elderly as a routine test. We just cannot do that because the disease starts decades before we see it in the clinic. We still have not found any treatments to stabilize the disease, to reverse the processes, and we definitely don't want to catch it when you see this frank hippocampal atrophy. As I heard in one of the presentations earlier this week, it almost feels like a sci-fi movie to start and reversing it at that stage. So one thing that is special to UW ADRC and was the pioneer was to use precision medicine approaches to look for a cure for AD. And it came from the use and success of precision medicine in cancer. And we are very far from it. What we can do right now is to look at disease modification and see how we can find people who are in the mild and moderate stages of the disease, what really is happening in their brains, and can we then design therapeutics. So it's still at modification, not at treatment. Just as the vignette, this is from the UK Alzheimer's Research Group. In roughly in a year, you have about 3,000 drugs that are developed based on precision medicine approaches. And only 2% were for dementia. None of them succeeded, but look at the contrast between dementia and cancer. And we see that with cancer, it has been relatively easy. So one of the things that I want to touch upon and kind of highlight is that AD, although yes, we have the two characteristic hallmarks, it is very complex as a disease, and it affects many, many biological pathways. These are some of them, not all. They're not necessarily independent, but they are different. So you have amyloid, tau, perfusion, vessel changes, changes in blood-brain barrier, immune response, lifestyle, that would include diet, sleep, exercise. You have genetics, and Alzheimer's disease seldom comes by itself.
Going further away.
So MRI can help understand these aspects. And I'm very excited to present to you these aspects because I love MRI. Before I start talking about the imaging that is done at ADRC, allow me to go over some of the things that Tom already went over. And these are not things done at ADRC, our ADRC, but it kind of reinforced what MRI is really useful for, and that is hippocampal atrophy. We do see that it is the greatest in Alzheimer's disease. This is a T1 image, and you can see that we can segment out the hippocampus and measure how big it is. If you look at an AUC curve and calculate accuracy, you'll see that in order to differentiate controls from Alzheimer's disease, the accuracy is 80%. What we are trying to do is capture people at an earlier stage, which we commonly refer as MCI. And at that time, if you try to find people who may be stable and who may actually progress to AD, our accuracy drops down quite a bit. This is another study that showed that E4, which is a genetic risk factor for Alzheimer's disease, it has one of the highest penetrance for Alzheimer's disease. You see that participants who have E4 alleles and who develop Alzheimer's disease have an even faster rate of hippocampal loss. So as I go forth through the first slides in the next, what I have done is I've tried to list some of the mechanisms that could be involved for that MRI technique. And I have listed the MRI technique in the corner. I apologize if that makes the slide too busy, but I thought it would give an idea of how MRI is being used, especially because it's not diagnostic and it is not really useful in AD processes per se. So this is hippocampal atrophy that we see with MRI. We do know that it is very strongly associated with CSF tau, which is a biomarker of AD. It does not relate to amyloid plaque deposition. So this is a very well-known fact, and we could do it in vivo, did not have to wait for pathology with MRI. This is also something that Tom showed, that we have these signature regions that are typically atrophying in Alzheimer's disease. Not everybody follows the pattern, which is why we have that topographic variance that Tom showed, but this is typically the pattern that we see. And this is all in the cortex, the gray matter and hippocampus volume. So what questions are we working on at the UW ADRC? So one question is, what are the other markers for AD? And one of the prominent markers that came up was NFL, which is neurofilament light chain. Just like we have amyloid and tau markers in PET, and we have corresponding CSF markers, we have a CSF marker for NFL. It is a marker associated with large myelinated fibers. And what we wanted to see is, we see all this cortical atrophy. Can we predict this atrophy earlier in time? And so this is a study not done on the ADRC cohort, it's done in ADNI. But what we found was that if you have baseline values of NFL, it could predict cortical atrophy in this frontal regions of the brain over time, over almost 2.5, 3 years. This is what the atrophy pattern would look like. And this is not an AD pattern, it's a different pattern. The second thing we could do is, we took that cortical thickness data and we said, well, if we add more information about Alzheimer's disease, we know that age is a risk factor. We know that amyloid beta and tau are important for diagnosis of AD. So let's take those, combine them with the MRI, and see if we can predict who is at risk of Alzheimer's and who is not. And our accuracy is still 65%. But these are some of the attempts that we made with the structural MRI, and to take it a bit further than what is established so far. So far, what we looked at was the gray matter, or the cortical surface of the brain. What is happening inside in the white matter? One of the prominent features that we see commonly is white matter hyperintensities. This is actually not a feature of Alzheimer's disease, it's a feature of cerebrovascular disease. And like I said, Alzheimer's disease seldom occurs by itself. Vascular disease is very, very prevalent in the elderly. What these are just white matter infarcts that you can see on a T2 flare. So I added an imaging component here. We don't see them in pathology. So if you take a brain and slice it up, you will not see this hyperintensity. It's only visible on this T2 flare. We do know that it correlates with cognition. We do know that it's highly prevalent. And it does relate to a lot of things that I've tried to list in the side. It is related to lower perfusion, diet, lipids. It's related to genetic risk factors, exercise. So how does this relate to what we know about MRI from a cortical and a hippocampal atrophy point of view? So something we did is look at the flare image, which is the second row here. And there's a common belief that the hyperintensities that are right around the ventricles have different mechanisms than those that are further away. The ones around the ventricles are called as periventricular. They're shown here in yellow, and they have more degenerative pathology and some venous pathology. As you go further away, the green ones, the deep white matter hyperintensities, those are the ones that have more of a vascular pathology. This MRI doesn't tell us that, but this was a start to looking at white matter hyperintensities. And then we looked at a lobar distribution in a small population. So this is just 33 controls, 25 MCI, not too many, but we do see a higher burden of frontal white matter hyperintensities in MCI population compared to the controls. For deep, we do see a much higher burden in the frontal cortex and also in the parietal cortex. Now, mechanistically, we think that the deep white matter hyperintensity burden is dependent on where you have the vascular tree branches and where the watershed zones are. So the two lobes kind of make sense in that in that context, but this still doesn't get us to a mechanism. It just gives you one more flavor of what pathology looks like in Alzheimer's plus vascular disease. A question that we often got from the ADRC was, well, how are these white matter hyperintensities related to that cortical atrophy pattern? How are they worsen cognition, but how? Because we just don't know what causes them. And so here, what we did is we looked at different regions of the brain and how they are connected to each other. So along the rows, you have different regions. For example, thalamus, superior parietal region, left side and right side of the thalamus, middle temporal lobe, caudate, so on and so on. And we looked at the connectivity of each one with each region with the other. And what this graph shows here is the neuronal fiber pathways that we can track using MRI by a technique called DTI. We tracked all these pathways through the brain. And we said, how many of these pathways are affected by white matter hyperintensities? And do they actually disrupt these pathways? So what we found was there were quite a few pathways that are affected by in MCI, and much more in MCI than controls. And that's kind of obvious from the previous slide because they do have a higher burden of white matter hyperintensities. This approach of looking at things also told us that this disruption in pathways is more lateralized. What we see is left-sided hemisphere corrections, right-sided hemisphere correction. There are very few connections that are left to right that are disrupted. So to put it in terms of a picture, I think that explains things better or easier is this is a 3D brain image, and these green, yellow, these colors represent different regions. And these blue dots here are the fibers terminating into these regions. And these specific blue areas are the areas that are affected by the white matter hyperintensities. So that nerve fiber pathway is disrupted because of the white matter hyperintensities. And you could imagine at some point down the line, we could ask the question, well, is there Valerian degeneration happening along the axon? That's why we see cortical atrophy, and it's actually related to degeneration due to these white matter hyperintensities. So we're starting to link mechanisms using different styles of MRI. To give a very specific example, if you look at the superior frontal cortex and the posterior cingulate cortex, this is two regions implicated in Alzheimer's disease. This is a T1 weighted scan. And in pink, you can see the white matter hyperintensities in this individual. In green, you see the pathway that connects the superior frontal cortex and the posterior cingulate. That's done through an MRI technique called DTI. And we see here that that pathway actually overlaps with the white matter hyperintensity. So if in a group of 53 individuals, when we look at the data, we found that there were 12 people in which this particular pathway was affected. In those people, we did find that their cortex was different. If this pathway was affected, their cortex was atrophied. Also, the people whose pathway was affected, that group had more people who had cognitive impairment. And so you could imagine this being taken forward to ask questions like, can we predict which cognitive domain is affected based on which pathways are affected? Can we support what the clinical core finds when they do their cognitive evaluations? And third, this is a comorbidity. This white matter hyperintensity is not an Alzheimer's feature. So how specific is it? Or is it just common and helps us look across all different types of dementia? So moving on, if white matter hyperintensities are indeed vascular disease, they're markers of vascular disease, how do they relate to perfusion and the vasculature? And so we look at blood flow and reactivity. And Tom mentioned this too. So this adds a couple more techniques that we look at, that is ASL and CVR BOLD. So ASL allows you to measure blood flow to the tissue, and it's quantitative, and it measures it in mils per 100 grams per minute. BOLD is a technique that helps you look at the changes in oxygenation. So normally you might have heard about fMRI, where you look at changes in oxygenation in the blood due to neuronal activity. But instead of a neuronal activity, you could actually make the person do a breath hold. So you increase carbon dioxide in the blood vessels. You could give them gases and change what they're inhaling in order to create this contrast. So in our first pass, we had participants who did the breath hold challenge. And here you see a group map showing that the entire cortex shows this MRI signal related to their breath hold pattern. And you see this is a blood flow map in all the individuals. In panel B, what you see is white matter hyperintensities. And this is actually a probability map. So the red areas show areas where almost every participant had a white matter hyperintensity. And blue is very few, maybe 1 or 2 had white matter hyperintensities. We looked inside the white matter hyperintensity, and then we looked a little bit outside the white matter hyperintensity to see what was the flow pattern like. And what you see is this reserve, this bold signal, is completely lost inside the white matter hyperintensity. The blood flow is halved inside the hyperintensity. And as you move away from that, there is no digital change. There is still a gradual, change in CVR and blood flow. So here is where you would think that the tissue is kind of normal, which is about, these numbers indicate millimeters. So about 10 millimeters away from the hyperintensity, you have normal blood flow, normal CVR. And it's starting to deplete as we come closer to the white matter hyperintensity. So this could we could ask the question, can we predict if the white matter hyperintensity grows into these areas of low CBF and low CVR? If so, can we prevent that extension of these white matter hyperintensities? And if we can find out that this is indeed a vascular cause, can we come up with some way of monitoring it, treating it, and find out which approach works best to not let these hyperintensities grow? This is again related to perfusion, vasculature, it is related to diet, immune response, exercise. There are large studies like the Sprint Mind study, that Framingham Heart study, which have shown that these hyperintensities play a key role in cognitive decline. And also, if you do manage things like blood pressure, you can reduce the growth of these hyperintensities. But the mechanism, what is actually happening in the brain, we haven't been able to relate that. And we think that approaches like ASL or CVR BOL would help us do it. So what the role does cerebrovascular pathology play in AD? I've been talking about cortical neurodegeneration, how it related to subcortical white matter injury, how that's related to vascular pathology. I'm kind of evolving the pathology and complicating it in some sense. So here, we're looking at CVR in about 100 participants, 91 participants. And we had their A beta 42 to p tau ratio in the CSF. So higher the ratio, more normal you are. Lower the ratio, worse is your AD pathology. And what this shows is that lower that ratio, lower is the CVR. So CVR is that bold response to a breath hold. And what it relates to is the ability of the vessels to dilate, to increase blood flow when you need it. So when you do a breath hold and carbon dioxide increases, you want more oxygen. You want the vessels to have the capability to dilate. From pathology, we do know that blood vessels are damaged in Alzheimer's disease. They do develop some calcifications, injury, so they may not be able to, they may not be as compliant as vessels in a healthy person. So this is an MR approach to look at that ability. We can see that that is also related to cognition. Lower MoCA scores in controls were related to lower CVR. In MCI, there was no correlation. So this mechanism of connecting physiology to cognition is just completely lost. This, I put it here for the sake of completion. This shows the same thing as the CVR. It shows how blood flow is related to AD pathology. And it is a similar pattern where if you have higher AD pathology, you have lower blood flow. But the reason I put uncorrected is because this, statistically, this observation was not significant. And so I would take that with a grain of salt, but I put it there for completion. And this is, if you look at this, it is only looking at vasculature and AD pathology. So yes, no diagnostic markers, but this is very useful in understanding what is happening in the brain. So we've been looking at perfusion, and this is all related to small vessels, as we refer it. We're looking at white matter tissue, gray matter tissue. What about the actual vessels? And so Dr. Chun Yuan, who directs the second research scanner, which is at South Lake Union, he looks at vessel geometry. And he has been working with a technique called MRA, or MR-angiography, that helps to automatically identify from the MRI scan different blood vessels and group them anatomically. And the question he wants to ask is, in Alzheimer's disease, how do these vessel structures look? How do their branching looks? Does it change over time? And is that really responsible for all the blood flow changes that we saw earlier? So these are some ways that we've already been looking at the ADRC and by the IB core members, like imaging and biomarker core members. But there are other ways to leverage the ADRC resources for MRI. And so this example is Dr. Angela Hansen's work. She has a project that looks at diet and how that affects Alzheimer's disease, how it actually interacts with E4 carriers. So she's looking at lipid tolerance tests, and she looks at blood flow changes after having a lipid drink for 4 hours after that. So the patient participants come in, we scan them for blood flow, then they drink their lipid, she scans them 1 hour later, 2 hours later, 3 hours later. And her reasoning behind doing that is she's using ASL, which is the technique to measure blood flow, as a marker for metabolism. And lipid metabolism is known to be, like a high-fat diet is known to be a risk for Alzheimer's disease. Lipid metabolism is one of the things in which E4 is involved. So she's trying to combine genetics, diet, lifestyle, and imaging to answer a complicated question. And we see this pattern where once you drink the lipid, the blood flow does decrease. This is a work in progress, and she's trying to figure out how this pattern looks different in E4 carriers versus non-carriers. This is another example. This is the pilot project. Tom briefly mentioned that the ADRC does provide opportunities for you to have projects with existing data or even new data. So this data that Hassam has is from the Stanford ADRC. And what he did here as part of his pilot project was to use the fMRI BOLD technique. And this is a technique to look at neuronal activity. So these are blood oxygen changes due to neuronal activity. He used it to look at a specific brain network called as default mode network, which is widely believed to be affected in Alzheimer's disease. And what he found is these regions, the medial prefrontal cortex, posterior cingulate, the lateral parietal regions, they kind of communicate with each other in the default mode network, but that communication pattern changes over time. And these are the 6 different stages in which he found the default mode network. And then he asked the question, does this temporal pattern of how the brain regions are talking to each other help us separate people who may have Alzheimer's pathology versus who do not? And with this approach, he got an 80% accuracy to classify Alzheimer's subjects from non-Alzheimer's. This is similar work that my RA, William Palmer, he extended it to look at the whole brain dynamics. And what he found is that in this experiment, this is ADNI data, if you look at all the different brain regions, they go through these 8 stages. And these stages are represented here in figure B. And what he showed is that in the MCI population, when you the brain continuously goes from one state to the other. It is not necessarily 1 to 2 to 3 to 4, but it is a pattern. And what he found is when you go from 3 to 4 and 4 to 3, that particular transition is very, very of very low probability in the MCI participants. And you can see this is stage 3, this is stage 4. They are different. The colorful nodes represent different networks. Red color means 2 regions are positively correlated with each other. Blue regions means they are negatively correlated with each other. And what he's showing here is the staging of stage 4 going to stage 3, stage 6 going to stage 3 are the ones that distinguish MCI participants from controls. And if you look at the stages, This stage 4 is default mode network and dorsal attention network. Sorry, for a frontoparietal network. And we do know that these two regions do communicate with each other and are involved in higher order cognitive processes. And what we are finding is that the MCI participants are stuck in this stage. They don't transition from this stage to 3. And this transition is what was critical in this case. So what I tried to show here was how MRI, even though not being diagnostic, can help answer many, many different questions. And it is not one technique, as opposed to PET, which is a much more expensive technique. It uses a radioactive tracer, so you can't really use it every time. MRI is relatively cheaper, much more easily accessible. And you have a laundry list of imaging modalities. We went through T1, T2, GRE, BOLD, task-based fMRI. We have CSF quantitative flow, ASL to look at blood-brain barrier. You can name a technique or you can name a mechanism, and we will try to find an imaging modality for you. That's what the imaging facilities, DISC and IBIC, do. It's like the Verizon app, you know, there's an app for that, something like that they say. But yes, yes, last night I actually tried to sit down and calculate how many MRI methods I have, how many mechanisms in AD I know, and how many possibilities there are. And then at midnight, I finally concluded it's just endless possibilities. So all these possibilities are open to you to try out with the ADRC. I hope I have excited you enough to try MR and to use all the resources that Tom outlined, and the imaging and the processing pieces that I showed through this presentation. So very briefly, adding on to what Tom showed, if you want to start an imaging study at the UW ADRC, you can reach out to me or Tom to discuss the project. If imaging is needed, then we usually have a project associated with the ADRC or a grant, budget numbers, IRBs, these are kind of the logistical details we need. We will make you fill out forms, and then we'll design the imaging protocols you need. So for example, we've had a study come to us and say, we want to see the effect of diesel fumes in participants. So we're going to replicate pollution. Can you image that? What techniques would be useful? And so we sit down and ask, well, what mechanism do you want to test? And then we'll come up with the MRI methods for that. So that's what we do. Then we'll go and look, train you for safety so that you can enter the MRI suite. Once all these things are in place, you can start scanning. Let's say you've done your scanning, or you have an existing imaging resource, one of the 4 or 5 resources that Tom pointed out, and you have a new question and you want your data analyzed. You can still do the same thing. You reach out to us. We can discuss processing needs, which method, what the goal is. We'll find you the right personnel to do the processing for you. We can train you. We can train an RA to do that. And we can provide the computational resources and neuroimaging software. So this is how DISC and IBIC work together as part of ADRC. We are in the medical center. We're not at Harborview. We're tucked away in the basement. And even though it may appear like we are sitting on the computer cranking numbers, it's a very dynamic group, so you should come and visit us. These are all the people who have contributed to the imaging results that you have seen, especially Tom, Kimi, Hassam, Dan, Jason. I think Jason is on the call. Annika is on the call. Our technologist, Tim, our assistant, Lisa, our RAs. Gray are members of the lab who are no longer here. Of course, our new shining scanner, the Engineer Edition, which we all fondly call it Elite Edition. We don't know what Edition stands for, but that's our interpretation. And then the participants who are willing to come in and get imaged with us for sometimes for about an hour and a half, and they're sitting still inside it too. So with that, I will stop sharing.
Okay.
Take question.
Thank you, Dr. Rane and Grabowski, for those talks. We're going to open it up for Q&A. Looks like we've got about 10 questions so far in the chat. If Feel free to toss in more things as more occur to you. I'm going to go ahead and stop recording.